The first one was about the efficacy and the safety of BI 1482-694. That is a third generation EGFR TKI with activity, with a specific activity against T790 positive tumors. And the investigators report about the results of a phase 2 study that showed in a sufficient number of patients that 62% of those patients were responsive to the treatment, to the single agent treatment. And there is, however, a caveat, because the confirmed overall response rate was down to 42%. Overall, the toxicity was manageable, and this agent should be considered as a treatment opportunity in the context of other available agents in the same class, like, for instance, osimertinib and ruxolitinib. Well, there are other new agents in the same class that are relatively late in the phase of development. Well, the question here is how to, having multiple agents, having first generation, second generation, now third generation EGFR TKI, how to sequence the treatment. Well, Dr. Tony Mock, in a subsequent discussion, was posing the question to use frontline, the third generation EGFR TKI, or to use first and second generation EGFR TKI, followed at the time of progression by the third generation EGFR TKI. This is still a pending question, and we are eager to see the results of the phase 3 studies that are ongoing in frontline, comparing the third generation EGFR TKI versus the first or second generation EGFR TKI. The other important issue is that there is an emerging evidence that following the third generation EGFR TKI, there are new mutations coming up. And this is leading to a more complex picture, because we need to define the right sequence of the drugs to be administered. And there is also a role for chemotherapy for some of these secondary or tertiary mutations, as you may consider those new mutations.